To remind you about, oh, sorry.
2 minutes to go.
So for folks joining the call, we'll kick off in a couple of minutes. We're just going to wait for everyone to make their way into the Zoom call. But yeah, please just hang on and we'll kick off soon.
Hi, Brad.
Morning, Tussie.
Good morning. So the
Morning, all.
handing over timekeeping to you.
Sure.
Thanks.
How long it talks?
25 minutes for the keynote speakers.
Yep.
Lightning talks 5 minutes.
Cool.
And it's currently recording.
Thanks, James.
I will clean.
Good.
We missed you yesterday, Brad, but James
I
did
know.
a good job. James did a good job.
Oh, good work, James.
I can never replace Brad though.
Got my bowl back.
Excellent. All right, it's 1 minute past the hour, so I think it's probably a good time to kick off this morning's session. So I just want to start by welcoming everybody to the third session of the Virtual Podium Asia Pacific for 2021. It's great to have you with us. My name is James Broadbent, and I'm a team leader of the CSIRO here in Brisbane in Australia. And you know, today the Virtual Podium Asia Pacific is about bringing together people across the regions, across the world, to connect, because we haven't had that same sort of opportunity in the last couple of years. So we're really lucky to have these sorts of things. We're really lucky to have a great committee to organise it, but particularly our chair, Tussie Rapasinghe, who's done such a great job of bringing people together, organising all of this. So a shout out to you there, Tussie. Thanks for all of your effort. And look, we're also really lucky to have sponsorship today. So we have sponsorship from Wiley, Rapid Communications and Mass Spectrometry. It's a great fit for the conference. We're going to hear a lot about mass spectrometry today and all of its types of applications across the omics. So thank you very much to Wiley for sponsoring, and they're sponsoring the Lightning Talks Awards that are really there for our early and mid-career researchers to show their research and get some more reach across the region and across the globe. So we're very happy to have all of you presenting today. So the format today, so we're going to have our two keynote speakers. So we'll start with Tianan, who's up first. Then we have a series of 4 presentations. So we have had a slight change to the schedule today, but we'll be managing that behind the scenes so no one has to worry. And then we'll wrap up with our second plenary speaker, Stephen Blanksby, at the end of the session. And hopefully there's lots of discussion. So there is a chat as well as a Q&A option as part of Zoom. So please jump on, post any questions, feel free to shout out as well when the time comes. So I'll be sharing the first lot of questions and Tulsi will be sharing later on. And also, you might have heard, so Brad Patterson's joined us this morning, so he's going to be doing some timekeeping. So if you're giving a lightning talk, Brad's going to let you know when you're sort of approaching that 5-minute mark. So please make sure that you keep an ear out for him. Don't be shocked and alarmed. Don't have to rush through, but just be aware. And so Brad's he's got his bell there, and he'll let you know. There you go. There's the sound to listen out for. Okay, so I think without any further ado, I'm going to hand over to Tianan Gyo. And so I met Tianan during his time in Australia, and I knew that he'd come from Rudy Aebersold's lab, so a really strong lab in mass spectrometry-based proteomics. And there's a heap of really great information up on the slide there, and I'm sure his reputation precedes him. But even when I first met Tianan, I could tell that he was a thought leader. He was all about, how can we get this quicker? How can we get it better? How can we get more sample numbers so that we can make better inferences, that we can apply all these really interesting new technologies like machine learning in that proteomics space? And it was really exciting to see him come through with all these really interesting ideas. And certainly since he's taken up his tenure at Westlake Institute for Advanced Sciences, the number of papers and interesting stuff that's come out of that lab has been phenomenal. So I'm so happy to have you here this morning, Tianan, and really excited to see what you've got to say. So I'm going to stop speaking. I'm going to pass it over to you, Tianan. So if you would like to share your slides, we can kick off with our first plenary presentation. Take it away, Tianan.
Yeah, thanks a lot for James for a very kind introduction. So without further delay, I'd like to introduce our recent research in developing a new mass spec data format. Actually, this can not only be used for proteomics, but can also be used for lipidomics, metabolomics, as long as the data is acquired in data independent acquisition mode, DIA mode. So let me introduce why we developed this new format and how it works. So as you know, today we can have multiple technologies to analyze different clinical specimens as well as other biological specimens. So our lab mainly focused on clinical proteomics. So recently we have a snapshot in Cell summarizing all the technologies for protein extraction and protein digestion, LC, mass spec for various types of samples. Solid tissues, cells, and liquid. And for the mass spectrometry, there are DDA, either label-free or labeled DDA, and targeted mass specs like SIM, PRM, or with peptides for absolute quantification. And the emerging technology is DIA mass spectrometry, and recently there are mobility integrated in DIA itself. So DIA can acquire all the fragments and all the precursor signal in a single digital archive, which can be later mined by different software. So this technology actually, together with increased throughput in sample preparation, largely reduced the cost for proteomic analysis. So earlier this year, we published a review in Advanced Drug Delivery Reviews. So let's assume the mass spec cost is $500 per hour, and this is based on the public data available in the internet in the US. I think this will be different for different countries, but then we can proportionally adjust the values as shown in these plots. So here we summarize the cost for measuring a single protein by mass spectrometry since 2006 till last year. So in 2006, the cost is about $3 per protein. And you can see it's linearly decreased until last year. It's about $0.1 per protein. This is thanks to the throughput of mass spec-based proteomics and also the depth, increased depth of proteomics. And I think this year or next year, the cost will further reduce. It's like what happened in the genomics field. Then the mass spec-based proteomics can be widely used in many areas, including clinical applications. So then it's increasingly being used in disease diagnosis and prognosis and for drug targets. And this built on the AI models, which are then rely on the omics data and the clinical data. So in omics data, multi-omics is a very hot topic, and the proteomics is in the center according to the biology principle, the central dogma. So the DRA actually is probably the most rapidly developed mass spec-based proteomics method in recent years. So we had a published review of DRA based proteomics, and basically this also can apply to metabolomics and lipidomics. And it's developed since 2003, but until recent years, it's widely used in various scenarios. And normally the way we use DIA is we collect, in clinical scenario, we collect samples from patients, we extract protein, peptides, we have DIA data, and then we use various software like Spectrum, Dye, open source, and we identify peptides and then peptides compared to proteins. And then for the protein matrix, we can do statistics or machine learning for phenotype prediction. So now in this talk, we present a different way. So from clinical samples, we rapidly generated DIA mass spec data, and then the DIA data directly convert into a format called DIA tensor. Tensor is a multi-dimensional matrix. And use this tensor, we can directly feed into deep learning for phenotype prediction without the need to identify peptide and proteins. Because if you want to identify peptides and proteins, then you are limited to those protein sequences that is in your dictionary, in a database. If there's for proteins like bisrabal, like the fusion protein in CML, then you will never be able to detect it. But with the DI tensor, we are not biased, and also the procedure will be much faster and more efficient. So in the next few slides, I will going to show how what is DI tensor and how it works. So the generation of DI tensor, we have the DI raw files from the vendor, can be from Sykes, Thermo, or Bruker, and they will centralize the data into MZ XML file, and then we read the file and read the attributes of the mass spectra, including the information shown here. Basically, they are M over Z, intensity, retention time, and so on. So normally there are some missing values. We need to generate a fully padded MS2 windows with the complete data set. And then it's optionally we can bin the MS2 spectra and then generate a DI tensor. So this figure shows for DI, normally we have different windows in multiple cycles, and they are for each window, we convert into this data matrix, and there are multiple windows, so they are multi-dimensional matrix. It's called a tensor. So this figure shows how it looks like in a swath window. So this window 1, window 2, and up to window 66. And green region here shows the window, because in each window there are some precursors not fully fragmented and they can easily visualized in this DIT formats. So compared to the WIF file in SWOS and MZ XML, the size of the DIT can be much lower in 1 or 2 orders magnitudes lower than SWOS, and also the same applies to the DI data generated in Orbitrap. So this video shows a video of the DIT we converted into time scale. So this is a SWOS window from the y-axis, which is the M/Z. The x-axis is the retention time. So each pixel indicates a fragment of the peptide from a particular protein, and scans from the lower M/Z to the high MZ range. We can also convert the DIT into a single image like this. This is from a scanning SWOS. So the SWOS window from the lower part to the higher part, and each lane here actually have a very condensed information content. If we randomly select 4 of them and we've rotated 90 degrees, then we can see there's a lot of information here. And then if we select small region and zoom in, we will see something like this. So it's like a universe, and there are a lot of stars, and each of these pixels indicates signal from a peptide or fragments. And we can see these eyes that are not randomly distributed. They form some clusters like galaxies in the universe. And we draw some lines to show this isolation of different regions in the proteome universe. So in a SWOS file of 45 minutes, and this DIT contains approximately 10 billion pixels, the huge amount of data. So we then developed a technology, actually a computational pipeline, to accommodate this DIT data and from a cohort, and then we can use the ResNet as one way of analyzing image data from the deep learning field, and this can be directly used to distinguish normal or tumor to diagnose tissues. We apply this to as a proof of principle to several exemplar data sets. One is liver tumors. So we have a training set containing 90 samples, and the test set containing only 12 samples. It's a limited amount. But through this tenfold cross-validation, we can achieve a relatively good ROC for separating tumor and benign. And if we use a matrix generated by OpenSWOS, the ROC is 0.86. If we use the matrix by DIT, the ROC is 0.94. We also apply this to several hundred thyroid tumors. They are benign, also malignant. Based on the discovery cohort and test cohort, we also get a better ROC for the DIT compared to protein matrices, and the details are summarized here. So this means if we use DIT, then the chance to separate different sample classes is actually outperform those with protein matrices. So recently we also further developed a DIT, and this is published to other DI format like DI-PASAP by integrating mobility and direct fusion shotgun protein analysis. This is a recent new data acquisition method by Joshua Cole, published in Nature Methods. We also apply this to scanning SWOS. This is scanning SWOS data. I showed this earlier. So, for example, for the DI-PASAP, basically the pipeline we established is based on the DI-PASAP.d files. We use TeamsPy to extract information. We can optionally bin the iMobility dimension of being M over Z dimension, and then we can have a bint MS spectral, and this can be fed into a new digital archive containing I-mobility, DIAT, and this can be subject to neural network analysis, also can convert into images, as you can see in last slides. So this is some visualization of the DIT for DI-PASAF. This is I-mobility dimension, and you can see here that we can easily visualize the DIT format for DI-PASAF. So we also apply this to a data set as published in Nature Communication last year, which are the urine proteome for COVID-19 samples and controls. So with this DIT format, we can divide a sample to training set and a validation set, and we applied multiple machine learning technologies to separate the COVID and long COVID. And I would say it looks quite reasonably good. But this is a very small data set. So now we are in our lab, we are further applying this method to much larger data set generated in our lab. So we recently also developed a pipeline to convert the DIT back to MZXML files for spectral interpretability. Because in previous slide we showed we can directly perform end-to-end prediction or classification based on DIT format, but we didn't know what proteins, what peptides are presented by the features which are distinguishing normal and benign COVID and non-COVID. So then we develop another pipeline. We can read the DIT file, we extract information and store the data, and then convert the MZ indexes back to real values and retrieve window RT values and generate MZXML. And this can be then searched by open source, by DINE, by other softwares. And this figure shows the proteins identified or peptides identified using the original MZXML and using the files converted back from DIAT, and they have a relatively good consistency. So last slide, I want to stress that now we have technologies that show first a few slides that we can generate proteome big data from clinical cohorts from various types of clinical specimens. And this data, particularly when they are in DIA format, can be very high throughput. So this high throughput data, if we use a conventional way to analyze them, it will be really challenging. The data size, data volume, and the computation will take a pretty long time to process. Now we have the DIT format is very small and very flexible and can be directly fed into neural network analysis. So this is something we are doing recently in our lab. So this is performed in Westlake University in our laboratory of proteome big data, and in collaboration with Professor Zhong Zhiluan's group in Beihang University and Stan Lee's group in Westlake University. And we also get support from Westlake Omics INC. And the work is mainly done by my PhD student, Fangfei Zhang. Okay, thank you.
Fantastic. That's a really great talk there, Tianan, and just really exciting stuff coming out of your lab all the time. And I really loved your analogy about the universe, looking at this sort of data and seeing these constellations. And they said that we know more about what's out in outer space than we do in our own bodies. So I think that's an excellent analogy just there. I can't see any questions in the chat, but I have a few, so I'm going to jump in and start asking. And one of the, I guess it's a bit controversial. Do we need to identify proteins anymore? Do we actually need to know what's going on, or do we just really need to, particularly when it comes to clinical questions, or can we just use classification algorithms? What do you think?
Yeah, currently the deep learning is entering clinic, and now they are using pathological images to identify, diagnose diseases. And conventionally, this is recognized by pathologists. They can see the cells and the structure of the cells, and then they tell the diagnosis. But now we don't know what features the AI is looking at when they read the image and then output a diagnosis. So I think the same analogy applies to proteomics. Now we have to use the expert software to identify proteins from mass spec spectrum, and based on the protein, we diagnose or we find the drug targets. But you can also use the raw data without any identification of protein. You can still tell whether this cancer is, whether this tumor is benign or malignant. I think in some case, it's probably not necessary for diagnosis. But if you want to find drug targets, you still need to know what protein it is.
Yeah, good point.
Then we identify the pipelines to interpret the BIT formats. This is but if we convert into raw data without the need to identify, one major benefit is you can largely shorten the gradient. So normally you need 1 hour or 30 minutes gradient to identify proteins, because you need to separate, you need to have the peak groups from DI files. But if you simply use the raw data, then there's no limitation. You can maybe use several seconds or several minutes gradient for a very complex protein. If the goal is just to identify is benign or malignant.
No, I can see you've already really thought about this stuff. You got an answer right there. I can see Fahana's got her hand up, so I might pass to you for a question.
Yeah, thank you, James, and thank you, Tianan. It was so great. I'm a metabolomics person. I don't have much idea of proteomics, but I can see that DIAMS is really good for discovery type of work. But my question is around pathway analysis. When we think about multiomics integration, can you use this type of data to do this?
Yes, we didn't show in the slides, but I'm not working on metabolomics, but we downloaded some metabolomic DI data, mass spec data, and we have convert them into DIT. And this works, this pipeline works, but we didn't have the complete data set to benchmark it. So in principle, as soon as the data can be converted into DIT, then there are multiple machine learning ways to integrate the mass spec proteomics or metabolic data, because in the deep learning, they just recognize the data format. They didn't know this is from a small molecule, lipids, or from peptides and proteins.
Thank you.
Thanks. And I can see there's another hand up there, so I might pass to Sri. He's got a question, and then Brad, are you after that?
No, I was just saying we're getting close to time if we're trying to keep on schedule.
Perfect. Thanks, mate. Okay, Sri, go for it.
And fantastic talk. It's incredible what you could do once you start integrating all the systems, right? I had it's kind of technical question, so I could be wrong in my understanding here, but in the current slide, what you're showing here, they're still being referred to as pixels, right? So is it one of those remnants from the computer image processing world where everything has to be converted back into RGB before they're crunched? Or the reason I'm bringing that up is it because also that kind of slows things down, because now we have all these really nice raw values that have to be converted back to an image and then treated as pixels before doing deep learning and machine learning on?
So there's an option to bin them, because the M over Z axis, they have mass accuracy. So in the retention time axis, there are also some sort of inaccuracy or variation from run to run. So in this software, we provide the option that you can bin, let's say for the triple top data from sites, we have a bin of 0.025 delta because there's the mass accuracy is about 550 ppm. So for the retention time, you can beam for every 1 second or every half a second. This option. So when you beam them, the file size significantly reduced, and the computational need will be largely accelerated. Yeah.
Sounds great. Thank you.
All right, and we're at time, so I just want to say thanks again, Tiana. Excellent talk. Really, really enjoyed it. So, in the interest of keeping on and keeping up with the schedule, Shri, you're up next. If you want to throw your slides up on the share,
I'll do that going right now.
Perfect. So Shri joins us from Tony Purcell's lab in Melbourne. And he sent through his slides last night, so I'm looking forward to seeing what he's put together. Thanks, Shri.
Can you see the right part of the screen?
Yeah, looks great.
Excellent. All right. Hi, all. Thank you so much for the opportunity to speak here today. So I'll just jump straight into it. Antigen-presenting cells scour your body, every corner of your body, looking for proteins that are not just of human origin, but also bacterial or viral origin, ingest them, and then chop them up into small pieces before being presented on the cell surface for recognition by CD4 positive T cells. And you can think of it like a Twitter service for the immune system, where at every moment, the cells are telling to the immune system what's happening at any given point in time. So if there's a viral infection happening within your body, there's likely a DC going over to the immune system and say, hey, look, there's a piece of virus here. Here's an example. There's a lot more virus where it came from. You got to go look at it. And this is how our immune system will then be engaged, activated to respond to this threat. It could be viral, bacterial, or tumor. And this is why it's important to understand what characteristics exist of these peptides. And another point to note is that even B cells need licensing from T cells when they present the right kind of antigen to actually generate the antibodies. So if you can think of it, it's a very central procedure for adaptive immunity to present antigen in this way. And we wanted to, we wanted to answer what characteristics of these peptides and can we actually characterize them enough to generate ideas about how to design better vaccines? An idea would be that we can generate the right kind of response. For example, maybe we can skew towards a B cell response or a T cell response. So to that end, we thought we'll do a fundamental analysis of the ligands being presented by a single cell line, sorry. So we took a BLCL, which is a B cell line, and then we analyzed the proteins from different HLA molecules of interest. So one of the unique aspects of this study is that we were able to look at a single cell line and then understand the interrelationship between these three HLA class II molecules. And we were able to pull down proteins using specific antibodies to HLA-DR, HLA-DQ, or HLA-DP. And then we separated out this peptide cargo and then analyzed them using mass spectrometry. In this instance, we studied them using a triple TOF or a thermo-Q exactive. So we identified around 71,000 endogenously processed HLA class II ligands from HLA-DR, DQ, and DP. And they matched what we already know about this. And that's always reassuring to know that we're pulling the right kinds of peptides and not contaminants, because they all peak around 15 to 14 mer length. And then the majority fall within 13 to 17 amino acids long. If you think about it, the binding groove of a HLA class II is open, so the peptide kind of cradles itself around it. And there you can see peptides of various lengths being presented in there. And that was good to know. And what was surprising, and something that hasn't been shown before, was that together, these three molecules increase the proteome available for immunosurveillance. So if you just look at the DP only, DQ only, or DR only, about how much of a protein is being covered for immune surveillance, there is not as much as how much they can contribute to together. And that means more of the proteome is available for immune surveillance, and that could be beneficial and offer us an evolutionary advantage to carry three different molecules doing the same thing. And that's also because they have different binding characteristics. And I don't have time to go into that, but that also is a fascinating aspect of it. And the last bit I wanted to touch on is that longer or more abundant proteins don't always contribute to more peptides. So what you're seeing on the x-axis here is the number of peptides per protein, and here is the protein length or protein abundance. And you would imagine if the length is getting longer, you would expect the number of peptides per protein to go up this way, and the same with abundance, but they don't actually go that way. And they are indeed non-significant if you do any statistical analysis on them. So with that, I would like to thank you all again for the opportunity, and we published this recently, and more about this is in the paper. And thank you again.
Fantastic. Thank you, Sri. And we are going to hold over questions to the end, and Tulsi will be chairing that session of questions. But next up, we have Geordie Nellis. So if you want to throw your slides up there, Geordie. So Geordie joined CSIRO in February of this year, and he's powered through one massive review that's under revision, and he's just about to hopefully publish his first original research paper. So very productive postdoc. Very happy to have him with the team here. I'll pass over to you, Geordie. Take it away.
Yeah, thank you very much for your introduction, James, and thanks to everybody for this opportunity to be here. So I'm gonna talk about food allergens, and specifically about peanut allergen, which unfortunately is still a major health burden, with about 2% of the Western population actually suffering from peanut allergy. And one of the big issues here is that food packaging regulations don't have any thresholds for at which point you need to report any cross-contamination or trace levels of an allergen in food. And this, of course, leads to companies always putting these may contain statements on packaging. So may contain peanut or may contain milk. And because these statements are everywhere, allergic people often take the risk and consume this food anyway, because otherwise there's so many things that they cannot eat. And it has actually been estimated specifically for peanuts that if we could get a 1 ppm labeling threshold for contamination, we could then protect about 99% of the allergic population from this type of allergic reactions. Unfortunately, the method that is currently used to quantify food allergens, ELISA, just has difficulties to be really accurate at that 1 ppm level due to matrix complexity, causing, of course, some cross-reactivity issues, but also due to food processing, which can cause hydrolysis or proteolysis and get that antibody recognition to get lost, basically. So targeted proteomics for this type of quantification might actually bring quite some nice advantages, like we won't have cross-reactivity issues because there's no issue with antibody recognition, and we can get that retention time, MS and MS data points to get less ambiguous results and increase food safety. However, the current methods that are being used for this have some issues with robustness, often producing different results in different food matrices. The analysis time is a bit too long, up to 20 hours, and that LOQ just doesn't cut that 1 ppm threshold I was talking about before. So we then actually set out to try and improve these issues to get a method that would would enable routine analysis for allergens with mass spec. So first of all, we looked at optimizing the robustness by trying different extraction buffers. So here are the 6 that we tried. I want particularly for you to look at the urea buffers and the TRIS one, which is the one that's mostly used in the literature. What we find here, so here I've extracted the same amounts of peanut from 2 different types of peanut butter, and then took a ratio of the areas under the peak for some proteotypic peptides for peanut allergens. And what you can clearly see is that this really gave skewed results. For one peanut butter much better than for another, while the ureas really went nicely to that one threshold. So that makes us believe that chaotropic agents are really much better. We found also, we confirmed these results in other matrices as well, but I don't have the time to show that here. We then tried to optimize speed. So we picked peptides for quantification that don't contain cysteine. So we looked if we could skip an alkylation step. And indeed, that worked quite well with similar errors and similar peak areas with or without alkylation. We then tried to look if we could shorten the digestion time, and if matrix assisted digestion as well as different temperatures could actually improve the digestion for certain peptides. So here's one of the peptides that was working well for peanuts. And as you can see, actually, if you do 1 hour at 37 degrees without any microwaving, you actually get a much higher signal than you would get for a standard overnight fast protocol, which is presented by the test line. This is, of course, not true for every peptide, but for some of the peptides that you're interested in for quantification, this worked really well. So we then have a method that doesn't need alkylation and that can be done in 1 hour, outperforming FASP. We then looked at that method with some validation. So we looked at the linear range, got a nice linear range from 0.24 to 1000 ppm with a good R-square for the 3 peptides that I showed. And if we looked at buffer versus matrix, so here we tried 2 different matrices, peanut muffin and peanut ice cream. In both cases, we got a bit of a matrix effect, but nothing that couldn't be corrected with matrix matched calibration. So in conclusion, this work improved the total analysis time from 6 to 20 hours to only about 2.5 hours, cutting the LOQ also about 5-fold, so now at 0.5 ppm, and reaching an acceptable recovery rate depending on the matrix, but between 73 and 106%. So this is what we then want to use for a multiplex assay for major food allergens. And I think my time's up. So thank you very much for your attention. And yeah, this was great.
Perfect. Thanks, Jordy. Really nice talk. So I have finished my chairing duties for today. So Rena, if you want to pop your slides up, I'm going to pass it to Tussi, who's going to introduce you and take us through to the end of the session.
Thanks, James. Yes, our next speaker, Arina from Edith Cowan University, Perth, Australia. Take away, Arina.
Thanks. Thank you for the opportunity, and also thanks for the introduction. My today's presentation includes some findings from the proteomic studies of lupin with a focus on the allergens from this seed. So to start with, let me introduce you to lupin. Lupin, it's a legume. It's coming from the same family as chickpeas, lentils, soybean, and peanut. It has unique nutritional profile, which is related to its high levels of protein and fiber. Also, many nutraceutical properties are attributed to lupins, which includes prevention of cardiovascular disease, type 2 diabetes, and obesity. However, lupin seeds are underutilized as human food and are mainly grown as green manure and animal feed. And one of the constraints is because lupin is an allergen and is subjected to mandatory allergen labeling on food products in many countries. So to study the lupin allergens and compare them across genetically diverse narrow-leaf lupin varieties, we have used this proteomics workflow. So first, the proteins were extracted from lupin flower, and then they were subjected to triptych digestion. The separation of the peptides was achieved using an expert nano-oil C415 system, and the SWOT acquisition was done on a 6600 SciEx mass spectrometer. Then we have used the lupin-specific database as an input in DIANN, which extracted the SWOT data for us. We did some QC checks on the data, some data curation, and this resulted in identification of over 3,000 proteins from these lupin varieties, and obviously quantitation of them as well. Then the proteins that they were quantified, their sequences were submitted into AllerCat Pro, which predicted the allergenicity potential of the proteins with the comparison it's making with the known allergens from different organisms, from different allergen databases. And this resulted in identification of number of putative allergens from lupin, which had high sequence homology with some reference allergens from lupin, soybean, chickpeas, peanuts, almonds, hazelnut, and pistachio. So this heat map is showing the level of sequence identity of the putative allergens with the reference allergens. These identified proteins, they belong to different protein families. They had different PFAM domains. Next, I would like to share the comparative results for some of the allergens that they were found most variables across the analyzed lines. So first, I have the BETV1 domain containing proteins. So the protein level data is normalized across the lines for easier comparison. As you can see, a couple of the lines have substantially higher levels of the pathogenesis-related proteins. Next, you see the oleosin profiles from lupin, which had high sequence homology with the known allergens from peanut, which belong to oleosin family. And even though the overall levels are quite similar across most of the lines, but I want to draw your attention to the profiles, which is quite distinct in the domesticated varieties of lupin. For instance, the protein label with the hot pink color here is more abundant across the domesticated varieties of lupin. Next is the delta congluten results. So this is one of the abundant proteins in lupin seeds. And as you can see, delta 2 isoform is not present in some of the lines. And finally, the most abundant proteins in lupin, beta conglutins, which are also known as the major allergens from this legume. So again, the overall levels of the beta conglutins are considerably lower in the domesticated varieties, and some of the beta congluten isoforms are not present in few of the domesticated varieties. So why is this important? Because these proteomics findings can benefit the selective breeding programs in order to develop hypoallergenic lupin varieties. Also, we were able to identify some distinct differences in the domesticated varieties in the sense of allergen profiles. So we are thinking about confirming this result also with doing some clinical testing on these lupin varieties. And also we will be applying these workflows to study the allergens in lupin food products in order to see how are the allergens changing as a result of food processing treatments. Thanks for your attention.
Thank you, Erin. Our next speaker is, I'll share my screen because Ramani Ko, she's from Western Sydney University, but unfortunately she won't be able to, so let me share the screen. And we'll play the video.
Hi everyone, I'm Ramani from the School of Medicine, Western Sydney University. The topic of my presentation is development of LC-MS method for the quantification of somatostatin in the whole blood. So the introduction about migraine, it's a common, complex and debilitating condition that affects 6 to 8% of males and 15 to 20% of females. And these are the characteristics of migraine. The graph on the left-hand side illustrates our hypothesis that the somatostatin levels in our blood, which is the blue line, vary throughout life in a manner which appear to mirror migraine incidence, the blue line. To test the correlation of somatostatin levels with migraine, we first need to know participation. And the mechanism of action, which is the right side image, somatostatin acts on the sending regulatory pathway, controlling how much information enters the brain. So it's a bit like a tap. During migraine, there is an information overload where normal sensory traffic perceived as painful, a hormonal brainstorm. So the current problems with measurements of somatostatin is the literature concentrations vary greatly from 2.5 picogram per ml to 195, and there's a huge variability within and across the study. So the current measurement is using VG-aminoassay, and we know that it has got some disadvantages like cross-reactivity, and then the usage of different antibody might give different results, and which can lead to false positive results. The challenges in development of the assay are the degradation of somatostatin is done by metalloproteases in the human blood, and this leads to the short half-life of somatostatin, which is 1 to 3 minutes. So the blood has to be collected and transported in a clinical environment so that the stability and integrity of somatostatin is maintained. So the methodology that I adopt for the essay is the pre-treatment of the whole blood and the treatment of whole blood. For the pre-treatment, I've optimized EDTA, protein, proline, isoleucine, and the tripeptide alanine, alanine, phenylalanine in plasma. And then the recovery is 98%. But then when I spike somatostatin in the whole blood, the recovery fell to 1%. So I had to repeat everything in the whole blood because the half-life of somatostatin is 2 to 3 minutes, as I mentioned earlier. So there might be a possibility that after isolating plasma from the blood, the somatostatin is getting degraded. So I optimized a concentration of formic acid, and then the recovery was approximately 60% every time. And then for the treatment of whole blood, I used protein precipitation, chloric phase extraction, and then lipid removal, drying of samples, and then reconstituting it, and then injecting it in LCMS. So for the conclusion, somatostatin concentrations of 2 nanogram per ml has been quantified in the whole blood, and this is the first study to show 100-fold differences in somatostatin measurements between whole blood and plasma. And the measures taken to describe the prevention of somatostatin metabolism in plasma are generally poorly described. Most are therefore likely to be ineffective in plasma and therefore can lead to gross underestimation in the whole blood. And we have successfully developed quantification method and have measured somatostatin levels at least 10 folds higher than previously reported. So that's it. For any questions, please shoot an email to this ID and I'll be happy to answer them. Thanks.
Thank you so much for all this keen lightning talk speak. So now the question time. Would like to get question for all the 4 speakers. For Ramanik, unfortunately, we have to send an email. So I'm just checking the chat. Just kicking off, can I ask question from Ani? Ani, sorry, I'm pronouncing your name wrong. The lupin content you are talking about mentioned, is there any relation to the taste with the different level of lupin? In have you ever correlate or any information? You mute.
Just on mute.
Sorry, correlation of the taste profiles with the protein profiles?
Yeah.
So at this stage, not yet. We had some targeted analysis on the congluteins, like the most abundant proteins, but that's something that we're planning to do later on. There are certain proteins that we know that like affect on the beanie flavor of lupin. But at this stage, we haven't analyzed those, but we have like sort of them in our SWOT data, or we can design some targeted analysis for specifically those.
Great. James, I think you raised the hand.
Yeah, thanks, Susie. I just had a question for you there, Shri. I was just thinking, you 31,000 peptides, it's quite a lot of numbers to work with. It's kind of in that machine learning space. Is the lab looking at doing any sort of machine learning with that information?
I think we are in touch with people who kind of try to, there are 2 aspects to it, right? So one aspect of machine learning could be applied to say, okay, if we understand characteristics of this, can we predict what kind of peptides can bind to these particular HLA molecules? And also another aspect would be, can they trigger a T cell response, which I think is a bit more trickier because we don't have enough information on that front, but we have enough information from the 21,000 to look at these peptides to predict bind, and actually it'll make the binding algorithms already available better because now we have much more richer data set, because previously some of these HLA molecules have hardly 100 or 150 or 200 peptides of which they were basing this on. So now we have thousands that'll refine this in future. Yeah.
I'll have to catch up with you offline. My postdoc's working on something similar, but not quite the same. So it'd be good to compare notes. Anyway, back to you there, Jesse.
Thanks, John.
I think Brad is raising hand. Brad.
Yeah, thanks. This one's for Geordie, just around the allergens. Do you think we'll ever get to a point where we have a universal sample preparation for all food types, or is that a bit of a long-term dream?
Yeah, that's a very good question. I think it's challenging for sure, but it depends on what you want to measure. So I think if you're interested in measuring quantitatively a few peptides that are indicative for allergens, maybe you can pick the right peptides that work for the same extraction protocol. So I think if you want to optimize for a proteome and you want to have the maximum amount of proteins identified, it'll be very difficult. But because if you have a nice set of different peptides that are all indicative for a certain allergen, it doesn't matter if you lose a few, as long as you have a good set that works in that protocol. I think we can move to that. Yeah.
Cool. And one more if I can, Tussi. I guess that 1 ppm mark, I have a profound ignorance over allergens and what that must be like. But is there a threshold for somebody who is, has an anaphylaxis reaction? Is there a known titration that they get?
Yeah, it's quite a complex procedure. So basically, what they do is people that volunteer for this kind of research that are allergic for allergens. So let's take peanut, for example. They get administered really small doses of allergen, and then slowly building up until they get a response. And then all that data gets used, and then you look statistically, if 99%, I think they put the cutoff at 99% of allergic people do not get any allergic response to that doses, then we consider that as a safe level. So that's
cool.
That's how those measurements are created.
I assume that's the same for gluten as well.
Yeah, it won't be 1 ppm. It'll be higher for gluten. I think it's 20 ppm or something on the top of my head.
Okay, thank you.
Thanks, Brad. Thanks, Jody. Can I ask one question from Sri? Sri, what is your favorite data processing workflow? You shown a very nice data. What's your best go-to data processing workflow?
So for the HLA peptides, we rely a lot on the Peaks software from the bioinformatics solutions, only because it does incredibly well with non-tryptic peptides. There are lovely search engines available from almost every vendor, right? But they kind of do a really good job with tryptic peptides or chymotrypsin or anything which have a good enzymatic digestion. But where we don't know what the ends will look like, they suffer a lot in terms of their performance. We've used Protein Pilot in the past. That also is actually quite okay with this, but Peaks outperforms them all in our hands, at least for now. These things change every year, as you know, and for now it's Peaks and we're academic. So whichever the next big, nice thing, we'll jump onto it.
Thank you. So, any questions from the participant? Anyone? I don't see any question in the chat. Pana, do you see any questions from the Q&A?
There is no question, but I can see James's hand is still raised.
Yeah, we got another 4 minutes.
I can ask another one while other folks think of something. But Sri, it's for you again, so you can jump off here. I've seen a lot of buzz around MS Fragger. I used it a long time ago. I think it's been upgraded a fair bit. So have you had a look at that? I guess it's not going to work if things need de novo sequencing if they're not in the genome. But yeah, any look at that so far?
Actually, I do like MS Fragger. I do like MS Fraga. The performance still, I think I haven't evaluated the most recent version, but at least the version before, it does really well. Like probably, for triptychs and stuff, that's really fast. For HLA peptides, again, it'll depend a lot. If you have good amount of memory on your system, you can get away with also doing a HLA search using that. But I think we're still evaluating it. It's not quite there yet with peaks, but we do occasionally get in touch with Alexei and have a chat, and that's kind of also ongoing at the moment. To where I think, watch this space, I guess.
Thanks, Sri. That's it for me. Back to you, Susi.
No. Thank you so much. I think it's a very nice coverage of proteomics, and so it's my great pleasure to introduce our next keynote speaker. I'll share my screen again. So. Home screen. So it's my great pleasure to introduce Professor Steve Blanksby. I think I knew Steve about more than 15 years, met at Melbourne Uni while you are working in Wollongong University. Now Steve is the Pro Vice Chancellor and Principal Research Fellow in Central Analytical Research Facility, which is in Queensland University of Technology. I think everyone in the world knows Steve's ozone ID with the lipid identification. It's a great information for anyone doing lipidomics. It's my great pleasure to introduce you, Steve. Over to you. I'll stop the sharing.
Thanks very much, Tusi, and a great opportunity. Very excited to have the opportunity to talk about some of the work that we've been doing, particularly over the last year or so, to develop methods for fatty acid discovery in particular. And so the title is inspired by the great man himself, Donald Rumsfeld. Donald was quoted some years ago, before there was another Donald, I guess, that took all our attention. This Donald kept our attention for a while. He had fantastic things to say, like this quote from 2002, where he said, "There are known knowns, there are things we know, and then there are, there are also things that are known unknowns. That is to say, we know there are some things we do not know, but there are also unknown unknowns. The ones we don't know. So if you bear with me for a sec, I actually think it's a really great exposition on what I think is the current state of lipidomics and probably the omics more generally. So what I would say is that there's things that we know, there's biomolecules that we know that that nature presents us with, and we know to some degree how it makes them. And we've got good techniques to detect them. So, they're the known knowns. And to some degree, we understand the limitations even of mass spectrometry to detect everything. So there are things that, biology and biochemistry are producing that we know that our particular method is not detecting very, very well. So suffice to say, there are things we know that we don't know, or we can't measure in the particular analysis that we're doing. But I guess what I've been more intrigued by recently is, how well do we actually understand the biology, and are there actually unknown unknowns where we know we're not seeing stuff in our analysis, and it's therefore not telling us that we're missing some understanding of the biology. So that's a fairly bold claim to make, but let me try and explain that a little bit more, and I'll stick to the context of lipidomics here. Most of what we understand from lipidomics is coming from mass spectrometry. And if you sort of plot out the lipids that we think we know on a mass to charge scale, and you sort of histogram them, most of the action is sort of in this 500 to 1,000. So there's a lot of molecular information that we're aware of. So that's the 40-odd thousand lipids that are in the lipid maps database. So our ability to actually identify each of those, and those would be, I guess, the known knowns, the things that could be there, is challenged because a lot of these molecules have either very similar mass or they have exactly the same mass. So as a consequence, even for the things we know about, there's going to be a sort of a dark lipidome, if you like, where our standard mass spectrometry approaches are going to let us down. And I guess there's this additional step where if we, there are still sitting behind that, the true unknown unknowns, all right, where we would be hoping if we could do our mass spectrometry better, we would make discoveries that would inform the biology and teach us new things about how cells are working. So let's look for a minute at the mass spectral data itself. It's kind of tempting to, you know, let the computer do it all, but it's occasionally good to actually have a look at the mass spectrum, and just, even if it's just to help us understand what our assignments and what our knowledge is predicated upon. So if this is a shotgun mass spectrum of a lipid extract, which it is an old rat brain extract that, as Tussi mentioned, was back from the days when I was at Wollongong, you spray it in, you have reasonable mass accuracy, and you can look at each of these peaks and it gives you some kind of readout of what lipid might be there. If you throw it against the lipid maps database, however, even if you've got that to that mass to charge to a relatively high degree of accuracy, you're still going to get multiple entries. And that's against, you know, they're the knowns, they're the things that are in the database that either someone's seen before or someone thought was wise to computationally generate based on known biology, if you like. So that's still 16 entries, right? So we still have these known unknowns, right? Based on just that mass to charge determination. So of course we do what all good mass spectrometrists would do. You would take that precursor ion and you would run a tandem mass spec, and sitting underneath that, you can use that to eliminate, draw lines through some of the possibilities in the database. And that would look a little bit like this, where you would say, okay, well, I see some fragments that are associated with this being a phosphatidylserine. I see some fragments in my tandem mass spectrum that are associated with the acyl chains, and one of those acyl chains by mass must be 18 carbons and fully saturated. One of them must be 18 carbons monounsaturated. So that allows me to exclude some things out of the database that have different structures. But then there's a whole range of other things that are in the database that actually there's no handle in my mass spectrum. There's no mass spectral information that would rule them in or out. And I guess the main one that I'm going to focus on today is the position of double bonds, right? So, you know, I have a monounsaturated chain. The double bond could be in a range of different positions. And you might think, just conventionally from mammalian biology, this is a rat brain. So we could have the N9. So that would be the oleic acid if it was cis geometry. And you could have N7, which would be cisphascinate. It's another common fatty acid. So we've got no information to say, is it this compound, is it this compound, or is it both? Or indeed, are there other metabolites that would also share this same the same mass spectral information that we have up to this point? So that leaves us with some ambiguity about, well, biologically, what's happening here, right? Because if it's a cisphascinate, it's coming from one pathway in metabolism. If it's an oleic acid, it's coming from another pathway. And then it's anchored onto a phosphatidylserine. So that would be telling us something about where that metabolite is being used within the cell and what it might be being used for. So this was really, you know, this gap, if you like, this knowledge gap is really nicely illustrated from this paper from the Fent lab a couple of years ago, where they basically did the sort of two types of analysis one would conventionally do. So you do the sort of the shotgun lipidomics and you develop this really rich picture along the lines of which I've just described. And that tells you something about what's happening sort of with the membrane lipids and their metabolism. And then on the flip side, you hydrolyze everything and you do GCMS, and that tells you what fatty acids you've got. But the problem is you don't know which of these fatty acids are sitting with which of these phospholipids. So you don't know where things are happening in the cell. and you don't know the connection between, say, the structure of the membrane and the structure of the fatty acids that are carried by the membrane phospholipids. So there's kind of a disconnect, if you like, in the underlying biology. But perhaps more striking was that in this analysis, this quite conventional analysis of the fatty acids, the Fendt group actually discovered that there was a whole nother fatty acid, and a significant amount of it, which had a double bond at the N10 position. Now, that is an example, I guess, of an unknown unknown, right? That shouldn't be there, all right? The canonical view of cellular metabolism, the KEGG pathways, if you like, wouldn't get you there, all right? So it was an example and a bit of a wake-up call to us that we can't assume, on the one hand, that the biology is going to tell us exactly what we need to go looking for. There's this reverse pathway where we need to be able to use our mass spectrometry and our lipidomics pipelines to be open-minded enough to discover new metabolites, and then try and use that information to map backwards to how the biology might be working, how it might be being perturbed. So in our lab, we've been working on the technologies to do this. We need to go beyond just having sort of a conventional collision-induced, an accurate mass spectrum and a collision-induced dissociation mass spectrum. And because we're focused on the positions of unsaturation as one area where we need more information on the biology, then we've been using a technique, or developing a technique, called ozone-induced dissociation. So that works by a small modification to the mass spectrometer, where we use an ozone generator. We generate ozone external to the mass spec. And in this case, this is a LTQ Orbitrap Elite. We then take a little bit of that ozone and we tee that in to allow that into the ion trap, so that as ions are being trapped within the mass spectrometer, so they're mass selected, they're allowed to react with the ozone, that reacts with the double bond and gives us a mass spectral readout of the position of the double bond. And a lot of this instrumentation development was done by Dave Marshall in our lab. That's, of course, the most flattering photo I could find of Dave. So one of the reasons that we like using the ozone technique is because it gives you an entirely predictable fragmentation. So coming back to a monounsaturated fatty acid like this one, if the double bond's at the 9 position, I can just draw you these little chemical cartoons and predict the mass to charge of the product ions that I see. So again, from this idea of searching for true unknowns or unknown unknowns, it's quite attractive, because it doesn't matter where the double bond is, doesn't matter if it's something that's been reported before. I don't need a database spectrum. I can just try and assign those double bonds. But just to try and convince you of that, based on those predictions, I can then take some monounsaturated fatty acids, commercially available standards, and we run them on that Orbitrap Elite with the ozone running. And you can see I see exactly 2 mass to charge ratios in those spectra, each of which uniquely corresponds to the position of that single carbon-carbon double bond. So then when I start applying this to complex samples, so this is the equivalent mass spectrum of the equivalent precursor ion corresponding to a phosphatidylcholine with 34 carbons and 1 double bond. But now we're taking this of lipid extracts of a range of different prostate cancer cell lines, or prostate-derived cell lines. They're not all cancerous. What you can see is, instead of seeing 1 pair of ions, we actually see 1, maybe 2, and in some cases, this 3rd set of pair of ions. So there's 1 double bond, but 3 signals, up to 3 sets of signals. So that means that there's actually 3 different compounds there. So rather than this being 1 lipid, it's at least in some instances a mixture of 3. And what's striking is that this one in particular, shown in green, this is this N10 double bond position. This is the one that was reported by the Fendt group a couple of years ago. But here we're able to show this is not now just a GCMS analysis. This is showing that that N10 fatty acid is actually incorporated into the phosphatidylcholine. In fact, the most abundant phospholipid in these cells. And in fact, in some of these cell lines, like this PC3 cell line here, it's a significant contributor. All right, and I'll show you some data on that. We did the back of the envelope calculations in this cell line. that the phosphatidylcholine with the N10 double bond is something like 8 mole% of the membrane. So it's actually a major contributor. It's not just some byproduct that's ticking away there in the background. So we're having this massive biological variation between the cell lines. And why is it important to look for these unknown unknowns, I guess? And that's because if you look at just, I guess, the data that you get from a conventional analysis, so these are the sorts of pie charts that one would generate from different phospholipid compositions based on, you know, a fairly standard shotgun analysis, you can see that there's a reasonable degree of conservation across those pie charts. So, you know, at that sort of cursory level, when you're just sort of looking at the known knowns, then you might say biologically or in terms of the lipidome, there's not much happening. But then if you look at, you know, these positions of unsaturation and the sort of relative quantitation of those, there's massive changes. So here's this PC3 cell line that I was talking about before, where about 50% of the phosphatidylcholine 34-1 is this N10 double bond position, which according to the canonical metabolism just shouldn't be there. So it gets even more interesting than that. So when you start having this extra layer of information about these different isomers, you start to learn that the distribution of those isomers is different depending which lipids you're looking at. So for example, if we do the conventional fatty acid analysis, so I'm showing you on the right there, you know, this is the FAMES GCMS analysis, it's the same as the FENT group did, and we just repeated that on our samples. You can certainly detect the N10. It's interesting, probably the reason people didn't see it earlier is because it's not super well resolved from maleic acid, but you can see it if you go looking for it. So you can generate these sort of pie charts, and that's kind of the integration of all the fatty acids, I guess, in these cell lines. But then when you look at the individual molecular species that are carrying these fatty acids, you see that there's a very different distribution of those different fatty acid isomers. And in fact, beyond just the N10, N9, and N7, for different classes like the phosphatidylglycerol, there's even a small amount of N8, all right? Again, it's a double bond position that shouldn't be there based on canonical lipid metabolism. But because we have now a method that allows us to look at unknown unknowns, we can see this. We don't need a standard to see it. We can just see it based on the predictable fragmentation in the ozonolysis reaction. So it's sort of sending us back to the drawing board is that could we actually broaden out a method, particularly to look at the fatty acids, that would allow us to take a very deep dive into just the fatty acidome, if you like, where we would be completely agnostic as to what was there and just use the technology to give us the richest picture possible and allow us to identify as many fatty acids as possible. So to do that, what we're going, instead of doing sort of a hydrolysis of the fatty acids and then doing making fatty acid methyl esters, we decided to do an LCMS approach where we'd hydrolyze all the fatty acids, liberate them from the complex lipids, and then derivatize them with this AMPP derivative, which gives a really bright signal upon electrospray. And then if you sort of make these derivatives, do electrospray, you can do the ozonolysis, you can see the different standards, you get the really clear signals. And if you do have a mixture, you can get a set of signals for each of those fatty acids. So to do this in a sort of a non-targeted way and get as much coverage as possible, we elected to do this on a modified SYNAPT mass spectrometer where we'd take this mixture of fatty acids, we'd splay them out on reverse phase chromatography, and then run them through the SYNAPT mass spectrometer where the SYNAPT we'd modified to introduce ozone into the traveling wave cell. That allows us to do very fast ozonolysis on everything that goes through. So this is taking essentially these same fatty acids extracts that the lipid extracts from these same cell lines, doing hydrolysis on them. And we'd previously done the GCMS, that's what I'm showing you in the top corner there. And there was another group by Ferrari et al. Who looked at the same PC3 cell line, and they also pulled out some interesting things in the GC. But the challenge they have is that, you know, the traditional GCMS is not actually giving you any sort of de novo information on the structure. You're just having to sort of cross yourself and assign retention times and hope for the best. When we ran these as the AMPP derivatives with the LCMS method and the ozone on the SYNAPT, you can see this is the profile of all the 18-1 fatty acids that are present. That's the total ion, the precursor ion chromatogram. But underneath that, we get all these unique XICs based on at least 4 different isomers that are present. And because the XICs are unique to the double bond position, we can assign that. And so you see, we see 7, 9, and 10 really clearly, and we see a strong evidence for this N12. So that previously, again, not reported in these cell lines or elsewhere. So what we've been, based on that sort of preliminary data, we wanted to try and turn this into a fully automated workflow where you wouldn't have to know anything about ozone, you wouldn't have to know anything about how these things might fragment, and actually develop this really rich picture of what fatty acids might be present in the sample. So we've done this using a modified Skyline approach, where we, using Python scripts, build our own libraries of where, basically de novo double bond position based on any fatty acids that, any numbers of carbons that you like. And then we go looking for them in this OzID data. So if we run that again over the cell lines, now this is the much more detailed analysis, you can see that we pull out all sorts of fascinating things. So there's multiple XICs there that correspond to different fatty acid positions. And we get, the signal to noise is exquisite, so that we have a dynamic range that's, 2 to 3 orders of magnitude, where we can drill down to things with very high confidence that are isomers that represent, less than 1% of the total fatty acid pool. So when we do that, we can really clearly quantify N7, 9, and 10, but we also see the N12, but we're also starting to pull out things like, again, N13. That's something you wouldn't go looking for based on the known biology. And sitting underneath these are even small amounts of trans isomers, which are being generated and utilized within these cells. So this is a way to get a picture of, I guess, the unknown fatty acids, and actually sends you back to looking at the canonical metabolism and saying, how could I generate these fatty acids based on the enzyme systems that are there? And without sort of taking a deep dive into that, as we did in this publication, what we're able to look at is the relationship between the different substrates and the enzymes, and plot a new path that would allow you to get, under certain conditions, to these fatty acids. And even if you look at samples which are very well trodden, so things like human plasma, and you apply this method, you also start to see these unusual fatty acids are not just, the result of aberrant cancer metabolism. They're actually everywhere. It's just that you're not seeing them because we're not looking for them. Essentially, they sit in the unknown pool. So in an act of pure hubris, I've named this fatty acid after myself. Okay, so that's blank-speic acid. Nobody else had named it, so it's mine now. And that would be, it turns out, we've seen it in human plasma, 16 carbons, 1 double bond, and the double bond's at the N8 position. And you can see the signal to noise on these things. There's the 2, okay, it's a shoulder on the TIC of the much more abundant 16-1 isomers, but these are the XICs of the 2 unique marker ions for that double bond position. We can pull out the things that you are expecting to be there, pleasingly, even at low abundance. So, all your polyunsaturates, you get these beautiful spectra where you get 2 marker ions for each double bond. So all of this information is there with high robustness and really good signal to noise, so that when you actually come back to trying to compile a list of what we have high confidence is actually present there, you can see that there's 20 plus fatty acids that we're pulling out that haven't previously been reported in plasma. And I guess some, like the blank spike acid, haven't really been reported in mammalian biology before. So this is, I think, becoming a really exciting way for us to build a richer picture of what molecules are present. And it's important that we know what's present, because that informs our understanding of the biology. I've used up all my time, so I won't talk about photodissociation, and I'll just skip to talking about some conclusions. So I hope, if I don't leave you with any other impression, I leave you with this impression that lipidomics still has many unknown unknowns. So I think it would be good if people didn't think about it like it's a solved problem, and that if it's not in the lipid maps database, it doesn't exist. Actually, I think there's a lot of true discovery that still needs to be done. And there's a lot of work that needs to be done in new ion activation methods, or new mass spectrometry, new chromatography methods to allow us to see what's truly there. And almost anywhere where we use one of these new modalities and we point it at a biological mixture, we always see a mixture of isomers. So it's never just one thing. And what's happening with these isomers is a real indication of what's happening with the underlying metabolism. So I guess where we may like to go with this in the future is to sort of say, can we use those changes in those isomer ratios as a sort of very sensitive marker to changes in the underlying biology and potentially the onset of disease? A lot of people involved in this work, so really grateful. Particularly want to point out the work that Dave Marshall and Beric Pode have done in our lab. All the cancer work, it's been led by Ruben Young out of our lab, and Ruben's just got his thesis accepted in the last week, so that's very exciting for all of us. And Phil Menzel's a new postdoc who's done a lot of the work on setting up the data pipeline for the LC-OS-ID. We're really pleased to have collaboration with Waters on implementation of these techniques on the Synapse. So with that, I'll stop and thank you for listening.
Thank you so much, Steve. Very nice talk. So just waiting to see more questions. Okay, Brent already raised his hand.
Thanks, Steve. I hope blank spike acid doesn't turn out to be something nasty, like a marker for something horrible. But
I haven't even thought about it.
With the OS-ID, I know it's been around for years. What's the state of the art with that? Because I remember there being a thing about leaving it out on the benchtop and just reacting things before being introduced to the mass spec. Is that still viable, or did that sort of hit a dead end somewhere?
No, you can do it any way you like, but I mean, I think when doing benchtop ozonolysis is still a fantastic way to go. But when you get to something with this complexity, where you're really trying to drill down to look at lipid isomers, then you need the ability to mass select, so that you actually know you're really confident of what the precursor is. So you've pretty much got one isomer population at a time, and then you're looking at the double bond positions within that population. So the implementation on the Synapse works really well, because we don't, I guess you're asking about state of the art, we don't have to slow down the analysis. That's all run on sort of the native duty cycle of the instrument. So you run
in real time, yeah.
In real time. And I guess where we're working towards with this is it's developing a bit of an archive. You've effectively done an ozonolysis of everything over time. And so, if we miss something in the data pipeline and we need to come back and reinterrogate based on new information, then we can do that. So that's why we're excited about that. And that setup also has the ability to integrate with MALDI and DESI. So we did a MALDI paper with Ron Heeren's group in Maastricht earlier this year. Same implementation on a Synapse, works really nicely. So you can get you can look at how these double bonds are, how the metabolism is varying across a tissue, for example.
Okay, cool. Thanks.
Thanks, Steve. Thanks, Brad. Can I ask that question about blank spike acid? So interesting, Steve. And will we be able to see it in plant? Is it on
In where, sorry?
any plant tissues? So have you
Yeah, so what we're trying to do is there's no one source of truth. It's actually kind of tricky, right? So you think how do I know? Like, I can't be totally sure. It's possible that someone's reported this in a GC spectrum from a plant extract in the 50s, and we just don't know because there just aren't the databases, right? And even if there are the databases, they don't use a common sort of nomenclature or way to identify. They don't have unique identifiers. So we want to sort of work towards that. So at the moment, we're trying to get everything back to some kind of system where we can search the literature and pull out as much as we can as a part of this. Because I think what we're seeing now, and it's not just here, but other methods as well, lots of things getting reported, and it would be nice to try and link that together somehow. Well, first link it to whether it has been reported before, and it could be that it's been reported in a completely different organism, and try and make those links. They're important biologically as well. It could be, like you say, we're looking at plasma here. It could be that whoever's plasma this was had eaten something in particular that spiked the blank-speic acid up. That's perfectly reasonable. We just don't know. So we're working towards that, trying to build the database a little bit better.
Thank you. I think Tarangani is with OG. Okay, go for it.
Yeah, thank you, Stefan. And it's really nice presentation and so interesting. Yeah, as I have presented yesterday, I'm working on a biomarker identifying process, like project for a pregnancy identification. So we are moving towards lipidomic studies as well. And I'm wondering whether this method you were talking about is only for fatty acid identification. Is that like we are planning to do non-targeted lipidomics? We spoke with Tusi earlier, and yeah, we are planning for a non-targeted lipidomics. So is that, so we would like to have, as you talked, we would like to have many lipidins, as many as like new things with the physiological changes of the horse's pregnancy. So I was wondering whether this method is only for fatty acids or we can do for all lipidomics as a whole.
Yep. So, I mean, previously, I showed you a few complex lipid ozonolysis mass spectra, right? So that works. Those have been done, and we've done quite a few of those over the years, mostly in a targeted capacity, where you sort of mass select them and do the ozonolysis there. But I guess we're working towards trying to, then we went back to look at these fatty acids because we thought it was sort of challenging enough in itself. But I think the logic that we've sort of developed from this workflow will enable us to sort of step that back up to look at sort of non-targeted ozonolysis of complex lipids as well. I mean, it was kind of interesting listening to the talks early on, this whole idea that, what if I didn't have to look at the mass spectra, but I just treated this as a rich source of pattern generation? I mean, we could do that. We could do that tomorrow, right? You basically would run LC and do ozonolysis of everything without mass selecting anything. And it would develop an incredibly rich pattern. And that pattern would potentially be quite useful in terms of just pattern recognition, but you also might be able to back out some interesting biological trends in that as well. It would definitely generate a very rich data set. And at the moment, we wouldn't have the tools that would allow you to pull out the kind of information that I've just described with the fatty acids.
Thank you, Steve. James, over to you.
I see we just spat out time, so I'll keep it quick. But really nice talk, Steve. Really cool way to use Skyline to pull out that information, those known unknowns or unknown unknowns. But I've always thought about you as a structural lipid chemist, but you're sort of taking on the aura of a lipidomic scientist. How do you feel about that?
I think it's a bit of a misnomer, but it's very kind of you to say so. Yeah, I mean, it was really, in the work we've done with Ruben's thesis, it has been really interesting to have the ability to sort of circle back. So interestingly, we actually discovered that N10 fatty acid before the Fent group did. But because I was such a poor biologist, I was totally skeptical that we chased our tail thinking it was an aberration of the experiment. So what's been pleasing over the last few years is to develop a bit more confidence. And it is actually quite satisfying where you sort of say, okay, well, we're seeing this metabolite. If we go back and sort of silence this pathway, then maybe we'll make more of that. And, watching that has been quite pleasing. So we're certainly thinking a lot more about that and having more sort of letting the mass spectrometry do full circle and inform how we set up the biological experiment as well. But I'm a long way from being to being a lipidologist. I'm just still a lot to learn.
Thanks, Steve. Back to you, Tussie.
Thank you. I love to ask one more last question, Steve, burning question. Seeing that 18, 1 fatty acid, 7, 9, and 13 as a position and 10, why they are not seen 11th position?
Yes, I mean, it depends where you look. Yeah, so we have seen that in other places. I can send you that sort of map of the metabolism that we feel like we've kind of extended a little bit. I mean, it does all sort of still boil down to the same. We're not proposing different enzymes, right? So it's a question of what it's the unusual substrates for the same, or the non-canonical substrates for the same enzymes. So, you can find in that map a way to get through to N11 based on the same enzyme systems that we have, and we have seen evidence for it under certain conditions. Yeah.
Oh, that's great. Thank you so much, Steve. So it's pretty much
there's a hand up in the attendees section.
Yeah.
If you wanted to invite Saeed to ask the question, I'm not sure. But I'd just like to thank everyone for making my timekeeping duties really easy. I do have to go, so I'll see you all tomorrow.
Thanks, Brad.
Thanks,
Brad.
So the attendee, sorry, I didn't get the name properly. Do you want to ask the question, unmute or chat? We can type.
Yeah, Saeed, if you're on the call, take yourself off mute and feel free to ask a question.
Okay, can you type the question? Saeed?
See, you have to, I think if it's Ali, you have to give him access. So you can click on his name and unmute him. Yeah.
Sorry, I can't find your name.
In the participant, in the attendees.
Yeah. Side.
Yep.
Can you okay?
Hello, can you hear me?
Perfect.
Very
He's
well,
all
thank you.
from Canada.
Yes.
Sorry.
Hi, Farhana. Thank you. So this is Ali, Olds College, Edmonton, Alberta, Canada. It's been a great event. Thank you for putting this together, to all the hosts. Stephen, maybe more of a philosophical question than a technical question. I do metabolomics and I apply it to agricultural research more than doing other omics, but can you give us an understanding of what is the expected proportion of the unknown unknowns in proteomics relative to the known knowns and the known unknowns and the unknown knowns? And I guess the philosophical aspect of it is that if you were to identify something that is unknown, wouldn't that really be a known unknown rather than an unknown? Just to give it a twist at the end of the session. Thank you.
Thanks, Ali. That's really is a interesting philosophical position. I can't really speak for proteomics. I mean, I guess broadly speaking, how much are we seeing? I don't know. I really don't know. I think that if you want to sort of look at the data that I've shown you today, if you say that just, you know, and I'm only just talking about isomers based on double bond position, right? So that's just one degree with which mass spectrometry is struggling with lipid assignments and perhaps metabolite assignments. You know, there's the examples I showed you that instead of it being one thing, it's 3 things or 4 things or sometimes more. So, I mean, you could say that if someone's having a good day for doing lipidomics, they're probably getting 800 reasonably confident identification. So you might multiply that by 4 or 5. That's as good a guess as any. But I actually think that what we're learning is that the metabolism is capable of producing many different varied things, and they're not always there, but they, you know, it's capable. The system is capable of producing them. So, you know, I think there's quite a few unknown unknowns, but that I appreciate that's not a very satisfying answer. But again, also appreciate that with the metabolome more broadly, you can't just kind of map it back onto, you know, with the proteome. If you just park the post-translational modification and you just say this is what the DNA should be giving me, you at least know, you kind of know what 100% ought to look like. That I think that's less clear as far as the metabolism is concerned, because there's the interplay between the substrate and the enzyme systems, and it's complex. So I guess broadly speaking, what we're looking for is, you know, how can we extend what mass spectrometry can do, and it will primarily be mass spectrometry, to give me a richer picture of what's there. Like, can I get more data points about what the structure of this, or structures of these molecules are? Yeah, so I guess there's actually a great paper to your last point. If we're just riffing on the philosophy, there's a great paper which is called something like when does the apocryphal become canonical? Right? So sure, I mean, that's how science works, right? You discover something new, and for a while it's apocryphal. But eventually, hopefully, if you're right, it becomes canonical.
Perfect. That makes sense. Thank you.
Okay, thank you so much. I think we are coming to end of the session. Before that, it's an award ceremony. So I'll share the screen. Today, the sponsor is Rapid Communication in Mass Spectrometry, Wiley. So it's mainly you can submit the manuscript to mass spectrometry based manuscripts, and it's 2.49 is the it's quite good. So I need to go to the next slide. Sorry. So the award winning. Today, the award winner for the lightning talk is Sri Ramaratnam. Sri, congratulations.
Thank you.
We will be contact you from Wiley to get your award as a $250 US cash prize. So it will be, but all the speakers has done wonderful presentations, and I wish we can give awards to everyone. But also the very big thank you to 2 keynote speakers, Tianan and Steve. It's made a really beautiful presentations today. Thank you so much, and making this event very successful. So James, do you want to add something?
Just a note of thanks as well to all of our speakers, for Brad, for timekeeping. It's been a great session. But I can see we're over time, so that's all I have to say this morning. Thank you.
Thank you.
Thanks all.
Thank you.
Thank you.
Pussy, you want to just stay on at the end? And if you can stop recording.